we have Dr. Orlandi over here, Chief Science Officer, AOAC International. And before inviting Dr. Orlandi, I would like to introduce her over here. Dr. Palmer A. Orlandi is the Deputy Executive Director and Chief Science Officer at AOAC International and is responsible for overseeing the AOAC Research Institute, standards development activities, and the Proficiency Testing Program. In addition, he handles business development responsibilities to include developing strategic partnerships with the goal of advancing voluntary consensus standards, setting activities, and strengthening international relations. He has an extensive background in government relations, food science and safety, regulatory affairs, and public health in the federal government. Prior joining AOAC International, he served more than 22 years at the U.S. Food and Drug Administrator, most recently as CSO and Research Director in the Office of Food and Veterinary Medicine. So here I would invite her on screen to talk on AOAC International's initiatives, food authenticity and safety. So can we please have the AV of Dr. Palmer Orlandi over here?
Greetings, everyone. I'm Paul Orlandi, the Chief Science Officer and Deputy Executive Director here at AOAC International. And it's a pleasure to be involved in this virtual conference on food authenticity and food safety. To set the stage, I'd like to take the opportunity to discuss the Food Authenticity Methods program here at AOAC, which is now in its second year. Using the underlying theme of how consensus standards and official methods of analysis can be used to overcome the challenges facing food authenticity that has a direct bearing on food safety and public health, I'd like to provide a little background and justification for our analytical program, recap some of the accomplishments to date that the program has enjoyed, and then give some insights into where the program is headed. Naturally, I think it's best to start with just a couple of definitions. First off, what is food authenticity? Well, in a nutshell, food authenticity really is the assurance that raw ingredients purchased by the food manufacturer are accurately documented. Food authenticity also touches on public confidence. And food authenticity really is, in that regard, the assurance that products purchased by the consumer are safe and reflect the stated quality. The other side of the coin when we talk about food authenticity is economically motivated adulteration. And in this regard, we term EMA as a wide range of deliberate acts which are designed to misrepresent the authenticity and value of a food product without the purchaser's knowledge or even the manufacturer's knowledge for economic gain of the seller. Some of the adulterations of commodities can be categorized through the addition of non-authentic substances or the removal or replacement of authentic substances, some of which have a severe impact on public health and public safety. So why this focus on food adulteration and food authenticity? Just a quick scan of the Discernus Food Fraud Database really answers that question quite well. Just in these top 5 commodities that range from meats, spices, juices, olive oils, and dairy products, and particularly cow's milk, we see that there are a vast number of instances of recorded adulterations. To combat this and to ensure that there are watchdogs out there, the literature for analytical methods addressing either the adulterated substance or the authenticity of a particular matrix, we have seen over the years an exponential growth in the analytical tools able to address some of the authenticity and fraud needs. Along with this explosion of analytical methods, though, come an awful lot of challenges, particularly for regulators and manufacturers in gaining public trust. What are some of these challenges? Well, I listed just six of them right here that come to mind. And these include what are the appropriate methods of choice and what are the performance standards that are used to instill confidence in those methods. What about the breadth of applicability, the technology that can be used? I touched on this fourth one just a little while ago, the disparate performance requirements. Not all of these methods have been either validated or use the same type of performance requirements to fulfill the needs to combat authenticity and to ensure the applicability of a particular method. And then to cap it off, there are disparate regulatory requirements globally among regulatory agencies, among participating countries who are watching or watchdogs for EMA worldwide. And then there's the changing environment and the rapidly expanding scope of need and the approaches taken for food fraud. This brings us to what the role of consensus analytical standards can play in overcoming some of the analytical challenges we just touched on. Analytical standards serve to document the needs for an analytical method first and foremost, and it provides a detailed description of how that method must perform. It includes method acceptance criteria, and then it is an agreement of all stakeholders and subject matter experts that these standards should form the basis for methods used in a variety of instances. All of this fits the general description of what consensus really means. It is that general agreement. It is a group that is in solidarity to achieve a particular goal. And as many of you are aware who are present and listening and participating in this virtual conference, it is consensus analytical standards that forms the basis for AOAC and its renown worldwide. One thing that I didn't touch on in developing standards is creating this balance of perspectives. And from AOAC's perspective, this is an important tool in creating analytical performance standards, and in this case, to combat food fraud and to guarantee food authenticity. From this perspective, this balance of perspective creates an ability to draw consensus from as wide a range, a number of different stakeholders as possible. And this ranges from broad perspectives that include academia, government, to include regulatory agencies, industry partners, and other NGOs. There are those with specific perspectives, which include a variety of different industry perspectives, such as from the food and beverage organizations, those who develop reference standards and reference materials, technology providers, and then there are the regional stakeholders that encompass the globe. So the next question that we may want to ask ourselves is, how do we go about the process of marrying consensus and performance standards to meet the analytical challenges? As many of you are well aware, AOAC has become renowned for the use of and the development of the SMPR, known as the Standard Method Performance Requirement, publications that list essentially the performance standards that any method must meet to achieve its purpose. What is found in an SMPR? Well, the SMPR essentially is a roadmap that begins with the specific intended use of the method and ends in identifying all those areas that any particular method must be used for. In between, we start with including how the method should be applied, the definitions upon which any method must use, and then the nuts and bolts of the method or the prospective method are the performance requirements and targets that the method must meet. And lastly, the commodities from which the method must be able to uh, uh, serve to detect any particular analyte. In this example, this is an SNPR for allergenic targets. Naturally, when we're talking about food authenticity, there are different perspectives on targets. Is the target a particular adulterant, whether known or unknown, or is the analyte to be addressed actually a commodity to verify that its qualities do indeed meet the definition of authentic? Again, encompassing the intended use, the definitions upon which the method developer must follow, the performance standards for the particular analytes of note, and then the commodities upon which that method needs to perform well. Naturally, AOAC moves beyond the development of the SNPR to use this to adopt official methods of analysis by expert review panels. And naturally, both the SNPR that has been adopted and approved by our stakeholders are then published, and the changes and any status changes are then updated in another AOAC publication, the ILM, the Inside Laboratory Management magazine. Now, also, a lot of you are also aware that really this is just the starting point to meet the analytical challenges. There is naturally, as I just pointed out, the adoption of expert consensus and publications of SNPRs, but that's just the first step. The next subsequent steps that are equally or even more important are the call for methods, the review of those methods by expert review panels, and the publication and adoption as a first action official method of analysis, principally involving meeting criteria for a single lab validation exercise. Over a period of 1 or 2 years, that method is then monitored for its applicability, for its performance, and for its inclusion in a multi-lab study of multiple unknowns, and the results of which then are reviewed again for adoption as a final action official method of analysis. Again, those of you who have been aware of AOAC's processes over the year, none of these steps should be foreign to you. So this brings us to the Food Authenticity Methods program started in 2019, the objectives of which were to address the analytical needs for combating international and economically motivated food adulteration. I tried to highlight in an earlier slide the breadth of analytical methods that are now inundating the literature, either looking at targeted adulterated analytes or ways to address the authenticity of particular commodities, standards of which are very faint and are very few and far between. That really is the inception of why a food authenticity program within AOAC is extremely important, to create that foundation of performance standards to guard against unverifiable performance of methods out there in the literature, and to create a consensus of what is needed in a variety of commodities and adulterants for verifying authenticity and to guard against public health implications in food safety. Our approach is to use SNPR development for targeted testing and non-targeted testing of a variety of high priority commodities, and to also involve the use of emerging molecular method applications to meet the other needs that cannot be captured by chemical analysis of some of the high priority matrices. The other key to the foundation for our program is not only to develop performance standards for potential methods to meet the needs of both consumers and industry alike, but also to address a need for rapid response in the event of public health threats, some of which we have already seen in the past, particularly with the emergency surrounding melanin in the first decade of the 2000s. And so one of our other objectives is to look at how we can develop consensus around a guidance document for rapid method development in the event of an emergency. These are the objectives that serve the food authenticity method program. I talked about two particular ways to approach the program. These are distinct but complementary. This involves targeted analysis and non-targeted analysis. And what exactly do we mean when we talk about this? Targeted analysis really is the determination of known molecules, a starting point of which we know what that adulterant, that analyte is. And in the case of targeted, it requires that prior identification of the naturally, the other side of this complementary approach is non-targeted analysis. And the concept is to create a fingerprint for a particular matrices. What are the characteristics that make that commodity authentic? And once that baseline is developed for a particular commodity, compare new areas or new test ingredients to see what the degree of difference, is in that commodity. Now, naturally, there is this continuum of difference. Is the difference small enough that it may raise concerns, but may be attributed to yield, seasonality, region? Or is there a large difference? Is there something within that profile that stands out, that screams there is an adulterant in this particular commodity? So naturally, non-targeted testing analysis is actually looking for differences based on the definition of what makes that particular commodity authentic. It establishes that true breadth, that scope, that balance of where authenticity can be assured and non-authentic starts. And it highlights the importance of reference materials in establishing what that standard profile must be. This is an inherent challenge in non-targeted testing. We talked about the elements that are important in the development of a standard method performance requirement document. We then explored a little bit about the nature behind targeted testing and non-targeted testing. Now it's time to bring the two together to show that there are some distinct differences that can be found and have been developed in our first 6 SNPRs to date within either targeted testing and non-targeted testing, following the framework, the SNPR framework. And naturally, from a targeted testing perspective, the applicability is directed against a particular known and specific analyte. The SMP defines what that targeted analyte is in the matrices of interest, and also then follows that the performance requirements are based upon analytical range, accuracy, reproducibility, and repeatability. and are dependent on a known certified reference material or a standard reference material to base their conclusions on. Non-targeted testing, however, is centered on something a little bit different, and that is, to use the same word, um, determining that there is something different between an authentic sample and a test sample. And the basis of the performance is being able to define what authentic is for that particular commodity. And naturally, the complexities are going to be even greater on this side because any reference material or reference materials used to generate that reference profile have to be agreed upon. And so you can see that even though within the constraints of what an SNPR is, the approaches and the definitions are going to be different, whether we are talking about call for methods for targeted or non-targeted testing. So in 2019, the Food Authenticity Methods Development Program started with the following priorities. They involved looking at three of the most notable commodities that are susceptible to adulteration, and these include milk, honey, and olive oils. And we started from what was known. Now, naturally, each of the two working groups that were established, the targeted testing and the non-targeted testing working groups, came at it from different perspectives. For targeted testing, there were a variety of already known adulterants that had affected the authenticity of each of these three commodities. And these things are listed on the slides, as you can see below each of the commodities. The next approach for this group, however, was to assess the gaps in current performances of those methods already in place and to determine where consensus needed to be found for each of these commodities and each of the major recognized adulterants moving forward. From the perspective of the non-targeted testing working group, the idea was to take these commodities and determine how best to create a fingerprint, to create that profile of known authentic samples, and then begin to see whether the known adulterants could be used as a way to see how those profiles are affected and the limits of detection that would be required or that are possible to pick up these known adulterants within the profile of authentic samples. We did highlight some of the differences in the approaches taken by the targeted testing and non-targeted testing working groups within our program. We also highlighted some of those differences that would be evident within SNPRs that address particular commodities, particular adulterants, and what would be needed in a method based on the performance standards found in any SNPR. I also mentioned a little bit earlier on that though targeted testing and non-targeted testing approaches are rather distinct, I also mentioned that they are complementary. And what I'd like to at least highlight in this slide is to show exactly one example of how that complementarity would work. And that is starting from a particular profile generated through non-targeted testing that identifies a particular commodity as having something different in it based on the consensus fingerprint. Now, does that non-targeted testing method give some idea of what it might be? And can we glean information from that particular profile and that anomaly, for which we can then use to either one, identify what that target is, or two, begin to develop a means to extract more information so that we can identify it. And once that target is identified, then generate a new method that targets that particular analyte specifically in that particular matrix. So olive oil, milk products, and honeys were our first commodities. But there are a number of other commodities and adulterants that are just as important. And once laying the groundwork for the 6 SMPRs, which are now out there and have been approved and are in publication, to move forward of which. And in 2020, our advisory panel looked at additional high priority raw materials and finished products and are now looking into the categories of botanicals and spices. In addition to those first two working groups for targeted testing and non-targeted testing, we have expanded to create a third working group for molecular and genomic applications to not only stand on its own in ways of identifying adulterants and to verify authenticity, but also to complement the performance standards that are largely revolve around analytical chemistry methods to look at adulterations. Those are principally the applications that are envisioned to come from the first 6 SMPRs. And to serve our third objective in creating guidance documents, we are carrying on work for emergency response and developing a decision tree to combine the complementary nature of targeted testing and non-targeted testing approaches. And lastly, it is intuitively important that to develop the methods either for targeted testing or non-targeted testing or for molecular applications, reference and testing materials are extremely important. This goes back to serving the needs of defining what authentic means. We have already been working with our 2020 Food Authenticity Advisory Panel members. This approach for 2020 was fully funded through the generous contributions made by Abbott Nutrition, Bio-Rad, the Coca-Cola Company, Eurofin Scientific, Herbalife, Mars, SGS North America, the Tentamus Group, and Thermo Scientific. We hope to continue with their participation into 2021 and look forward to it. Now, just in closing, what are the benefits to being part of this program? And it's all encompassing in those areas that I touched on recently. We provide the consensus performance standard methods for commodities and adulterants alike in defining authenticity and in identifying unknown or known adulterants that jeopardize public health, that jeopardize food safety, and help to combat economically motivated adulteration. I'd like to thank you for allowing me to provide some insights into food adulteration from AOAC International. But before I close, I'd like to make sure that everyone is aware of the fine contributions we get from our AOAC volunteer leadership, particularly through our volunteer science advisor, Dr. Bert Popping, and our 3 working group chairs, Dr. Joe Boysen, who chairs the targeted analysis working group, Dr. John Spilka, who chairs the non-targeted analysis working group, and the newest addition, Dr. Danielle Soyer, who is chairing our molecular applications working group. With that, I'll close and leave you with a slide for contact information if you have any questions about the AOAC program. And I'd like to thank you very much and good day.